Ladies and gentlemen, back today, we've got somebody I love to have on the program because we're going to be talking about fungi. We've got Professor Xu Jianping from the University of McMaster up there in Canada, the Department of Biology. How are you doing today, my friend?
I'm doing very well. Thank you very much for having me back.
Yeah, it's always a pleasure to talk to you because I always learn something new, and that's the most fun. So we were actually just talking. You wrote, I just want to talk about these papers that you wrote just recently. There was some cases in Himalaya and the cases in India where we're facing certain kind of fungal pathogens. Is that right?
Yes, that's correct. So in the Indian case, was a rare fungal pathogen called Rhodomyces elongisporus. It's a rare yeast pathogen, but was found to cause an intensive care outbreak for neonates, for the new babies. And this study involved 10 new babies over a 6-month period. And all of those infecting strands were genetically highly related to each other.
Wow.
And surprisingly, some of those strains were resistant to bleach, to the disinfectant. So they were found to colonize the warmer, baby warmer, for example.
What are the go-to, is bleach, is that normal if you're trying to take care of a fungal pathogen? Is that something you use, and what else would you use?
Yeah, bleach is very commonly used as a surface disinfectant in both hospitals and even in typical households.
Yeah.
So typically in those situations, you try to use chemical methods or sometimes physical methods, like UV radiation quite often is also used.
So just a little sunlight would kill most of that.
Yeah, actually, sunlight, if you expose long enough, can kill a lot of things. Yes.
Okay, maybe that's why my mushrooms died that I put in my yard. So also, yeah, this story in Himalaya. Can you talk about this?
Sure. So I have been involved in. Actually, this is not published yet, but it's going to come out.
Spoiler alert.
Spoiler alert. So we had a paper accepted on Monday, and this paper basically described in a very, very remote area in eastern Himalaya, where we sampled over 300 strands in 17 different locations, very undeveloped area, very big rivers and deep rivers and high mountains surrounding that area. We isolated a strain of a specific fungus called Aspergillus fumigatus, which is a common mold, but it's an opportunistic human pathogen. And what we found was that about 7% of the 300 some isolates were resistant to the 2 commonly used antifungal drugs in the clinic for treating infection by this pathogen. So that means that when we are exposed to those infections, those drugs would not be effective. And this was a bit unexpected because this area was very isolated, very remote, and very few people live there. But still, the drug-resistant strains that probably developed somewhere else were able to disperse to those remote areas.
How do they do that? Can they go with the wind? Are they in the air?
Absolutely wonderful question. Yes, they can. So this fungus can produce many, many spores, and all those spores are very, very light. They can become airborne and then carried around by wind. Of course, it's also possible that some human activities like travel and the transport of goods in some of those areas, even just bypassing it, potentially could bring microorganisms from other areas to that area.
Yeah, check your suitcases before you travel, people, because you don't know what you could be traveling with. So as far as the drugs that they tried that they failed with, what are the 2 drugs that usually do well by fungal pathogens?
Yeah, so for this specific species, the 2 drugs commonly used first-line treatment. One is called the itraconazole, Another one is called voriconazole, two triazole drugs. And 7% of those strains were resistant to either one or both of the two drugs.
Wow. And so talk about the implications for the spread of something like this. Now, is this something that would be able to be spread human to human, or is this something that comes from the air and then people just get sick and die?
So this one is primarily spread from the environment to the human. We get the exposure from the environment. Most of the time when we are infected with this fungus, if your immunity is good, you would be able to clear the infection. If the immunity is not good, then the fungal pathogen will start colonizing and growing inside you.
Now, would it die when you died, or would it continue to kind of help decompose your biological remains?
It's going to continuously decompose the biological remains, and then it's a saprophyte, right? In that they can grow on that organic matter. So will become the substrate for them, continuously become the substrate for them, and then eventually it will be released. So in a way, it could be, if you consider that to be a human-to-human transmission, that would qualify. You could say that could be potentially transmitted from human to human.
Yeah. So what was that word you used? Simplified. What does that mean?
Oh, saprophyte.
Saprophyte. And entertain me here. I don't know what that is. Can you?
So that means they eat dead organic matter.
Oh, okay.
That means to survive on dead organic matter.
Well,
that's where the carbon and energy source.
That's us. We're humans. We're yeah.
Yeah.
Okay.
Yeah. So this fungus is very common in the compost pile as well, in organic debris.
Nice. So it's not bad. It's not a bad thing. So you also studied mitochondria, which is having to do with the DNA and stuff like this down on this molecular level. So what is mitochondrion? And I'm guessing that's the singular, and mitochondria is the plural. Okay, I'm learning. Can you explain a little bit about mitochondria?
Excellent. Yes. So mitochondria is an intracellular organelle, and it means it's an organelle is a structure inside a eukaryotic cell. And it was hypothesized to have originated billions of years ago through endosymbiosis, in that one cell eat another cell, and that cell that was eaten gradually turned into mitochondria. And it is very important for almost all eukaryotes, but not all. For almost all eukaryotes, is that this is the site where energy for cellular function is generated. So the universal energy source in all organisms is ATP. And in eukaryotes, ATP is generated primarily through mitochondria.
Uh-huh.
So this is like the powerhouse. This is the energy and the kind of generator for the community to power our house and power everything. That you can consider, if the community is a cell, then the power station is the mitochondria.
So where does it get its food from, or its energy from?
Yeah, so it gets its energy from the food we ingest. For example, for us, it's the food that we eat, the vegetables, the starch, the proteins, etc., that we eat, that converts into sugar, like glucose. And glucose then is taken up by the mitochondria. And it will undergo a series of chemical reactions, generate a proton gradient across the mitochondrial membrane, inner membrane, and that proton gradient then drives the ATP synthesis, converting ADP plus phosphate into ATP. And then ATP is used to power many of the other biochemical reactions.
Is it true that there are different metabolic reactions to these things, and like how they're used or how they're digested?
Yes, absolutely. So there are different reactions within the cell, and there are different substrates that could be used by the mitochondria that are imported into the mitochondria and then go through the chemical reactions within the mitochondria. And in fact, the mitochondria itself generate through those chemical reactions, it generates a lot of metabolites. And some of those metabolites are also used as substrates for synthesizing amino acids, for synthesizing nucleotides, etc., that we eventually use for DNA replication, for protein synthesis, etc., for cell renewal. So it's an integrated organelle with the rest of the cell.
Interesting. And so it's, would you say symbiotic or what? And you use the word endosymbiotic. What is that?
Endosymbiotic, yes. It means that it's inside another cell. They form a symbiotic relationship, form a mutually beneficial relationship. But now the mitochondria is an integral part of the eukaryotic cell.
Uh-huh. Interesting.
So when we move our muscle, when every time when we speak, when we walk around, we are powered by the ADP generated by the mitochondria. So it's very essential organelle for us.
All right. And do most creatures have it? Fish, mammals, birds, things like that?
Yep. Most eukaryotes have it. Eukaryotes means that they have a nuclear membrane around their genetic material.
Okay.
But prokaryotes, such as bacteria and archaea, they don't. They don't have mitochondria.
What were some examples of prokaryote?
Like E. coli, like bacillus. Those are bacteria. They don't have it. Those are called prokaryotes, in that their genetic material is not enclosed in a membrane, and they don't have mitochondria.
Well, they're very popular. I think they're getting by without it just fine.
Yes. So they have metabolic pathways that actually are very similar to the mitochondria, in that they can generate this proton gradient across their inner membrane and then use that to drive ADP synthesis.
Wow. Very cool.
And mitochondria's ancestry, the progenitor for mitochondria, is the bacteria.
Wow. And how old does it go back to?
Billions of years.
Yeah.
So hypothesized probably at least 1.5-ish billion years or maybe even more. The exact age is not really known,
but very close to the building blocks of life, though, right?
Yeah, not too far. Well, a bit after the origin of the first cell, the first life.
Wow.
And then, yes.
Which, who knows how that came? Maybe from space. We don't know. So what about mitochondria first struck you as being, like, obviously you like fungi, and we talked about that last time, but what was it about mitochondria that you find so interesting?
What I find mitochondria really interesting is that the inheritance pattern, and for both, we call it asexual reproduction and sexual reproduction, are very different from the typical genetic material in our nucleus. So let's just start with the case of humans. In humans, each of us have 23 pairs of chromosomes, right? One set is from our mother, another set is from our father. For females, you have 2 X chromosomes. You have 22 autosomes that pair with each other, and you also have the 2 X chromosomes. For male, we would have 22 autosomes, pairs of autosomes, and then 1 X chromosome and 1 Y chromosome. Okay, so those genetic material is stored in the nucleus, and then for the mitochondria, so those materials are basically half and half each inherited from the 2 parents, right? And then for the mitochondria, we're only inheriting our mitochondria from our mother.
Really?
Yes.
Is this new information?
I know. So this has been known for decades.
News to me.
So yeah, we get our for the male we get our X chromosome from mother, we get our mitochondria from our mother, and we get the Y chromosome from our father.
Interesting.
And the Y chromosome has relatively few genes, so most of the functional genes on the sex chromosome is from our mother. So genetically, we are much more similar to our mother. So what's striking is that you can see now the nuclear genome is contributed by both parents equally, relatively speaking. Okay, but the mitochondria, it's only from one parent. And we know that by combining genomes from both parents, we could potentially get the benefits. You have the good genes from the mother, good genes from the father, combined, make a very smart Chris Jeffrey. Okay, but the mitochondria, they don't do that for humans and for most of the animals and the plants. They don't do that. It's usually only inherited from one parent, and in the case of most animals, it's from the mother. So this has created this kind of evolutionary dilemma, because we know the benefits of combining genes, and we also know that if you do not combine genes and just inherit genetic material from one parent, there's potential through evolution that some of those genes in the mitochondria would accumulate bad mutations. We call it the deleterious mutations. And because you do not have good copies to fix it, potentially those DNA would get more and more degenerated and get less and less fit. And indeed, the less fit mitochondria has been associated with a lot of diseases in humans, including infertility, because in the process of fertilization, the sperm needs to swim, right, towards the egg. And you know what powers the sperm to swim? Mitochondria.
Yeah. And that's in the little swimmer. There's mitochondria.
Mitochondria primarily are in the tail of the sperm.
Okay.
The head is usually the nuclear material, the nuclear genetic material, and then the tail is flipping, and all those swim is due to the mitochondria in the tail, and it's driving that action. So imagine if you have mutation in the mitochondria genome, and it's not mutation makes mitochondria not able to generate as much energy. The viability or the fertility of the sperm would decrease.
Can there be mutations that actually improve the functionality of them?
Yeah, so it could be. So there's selection. When you have mutation in some of those genes, some of them may make them grow faster, and those swim faster, and those would be selected for in that next generation. Potentially, the DNA, the nuclear DNA associated with those mitochondrial mutation would be selected for.
Is it fair to say that when we get it right, it's evolution? When we get it wrong, it's a mutation?
Well, so mutation is the source of both good and bad. It's how they are selected.
Oh, okay.
What's been selected what's mostly we found in nature, it's been selected already. So those are the ones that are able to reproduce and are able to survive. And the ones that are really bad, for example, the sperm that have the mitochondria that's just not able to produce any energy, those sperm are dead. They're selected against. They cannot fertilize, cannot produce next generation.
Yeah. They just swim in circles and just give up. So, but you're saying mutation is just a word. It doesn't have the pejorative connotations that we put on it. Because when we say mutant, somebody goes, "Oh, well, that's mean." They called me a mutant all the time growing up. So.
Well, in many ways, we all are mutants.
Right. Yeah.
Here's a little bit of stats in that every time DNA replicates, there's a chance of mutation. So we have about 3 billion nucleotides in our DNA. Our genome size is about 3.1 times 10 to the 9th nucleotides. Every time it replicates, there's a possibility on average of a couple of mutations in our DNA.
What are some of the driving reasons why they mutate? I mean, obviously, toxic waste.
Yeah, so those toxic waste could increase the mutation rate, but the DNA replication machinery is not foolproof. It's we like to think that everything is foolproof inside our cells, but the DNA replication polymerase, it's not 100% foolproof. There's a chance every time we replicate, like 10 to the minus 9th nucleotide potentially would be a different nucleotides. So you have those kind of mutation. And in the presence of mutants, in presence of mutagens, like some nasty chemicals and the physical forces like UV radiation and the gamma radiation and the new exposure to high radiation, those increases the mutation rate drastically.
Wow.
Okay. So this is the kind of dilemma, the kind of dichotomy of uniparental inheritance for the mitochondria and biparental inheritance for the nuclear genome. So why is it so? How do we test the potential hypothesis about it? So in humans and in animals, we know that the sperm is much smaller than the egg. The egg is big. Okay. The sperm is very small. Small is just basically packaged a nucleus with mitochondria in the tail and trying to swim and fertilize the egg. And the egg would have a lot of cytoplasm, a big cell, and as soon as it's fertilized, they start to divide, etc. So one hypothesis was that, because the different size, the egg has a lot of mitochondria in there, the sperm has relatively few mitochondria, and all the mitochondria is in the tail. So after fertilization, very few mitochondria would be able to enter the egg. Therefore, we would have uniparental inheritance. The inheritance of the mitochondria from the mother, from the egg.
Yeah. I mean, when it cuts off the tail almost, it's so the head can get in, but then they lose the tail. Oh.
Yeah, lose the tail. Yeah. So this would be a very simple explanation, right? However, it has been found that even if some of those, sometimes the mitochondria from the sperm can get in there, get into the egg, but after getting in there, there's a mechanism to directly target them for degradation, and that they're selected against. There's a machinery selected against them.
Wow.
So why is it so? So because of this, in animals, there's this we call the anisogamy, in that the different sizes of the sperm and egg. It's very difficult. We cannot change that ratio, right? We cannot change the size of the gametes. So it's difficult to test hypothesis against that explanation. Then we thought about other systems. For example, in fungi, where quite often the gametes are not as differentiated as that in animals, in that the gametes are similar in size. But even in most of those systems, there seems to be a mechanism to maintain uniparental mitochondrial inheritance. So in the system that we work with, called the Cryptococcus neoformans, is a yeast, and there are two sexes, okay, in the system. Are you following me?
Yeah, because you're saying that no matter what you do, the women rule. The women get the is that right?
Yes. Yeah.
I love this. I'm taking notes.
Yeah.
I'm taking notes.
Yeah. Good. So your mitochondria is from your mom, mitochondria is from my mom, and my kids' mitochondria from my wife.
Women, right? Ah, geez.
Okay. So in fungi, in this yeast that I was starting to introduce you to, Cryptococcus neoformans, there are two sexes, just like male and female. And in this case, it's called A and alpha. And this yeast, you have, there's no difference between A and alpha in the cell size. Okay. They're identical. And when they mate, they diffuse.
Interesting.
And you would expect that the fusion product, the zygote, would have mitochondria from both parents, right? Yeah.
No,
they're not.
That's crazy. Even when it's equal,
even when it's equal,
the mitochondria only gets passed through. So what do we know about why?
Very good point. Very good question. So why is it only one of the parental mitochondria is inherited in the progeny for most eukaryotes? So many hypotheses have been proposed. One of the ones that was I mentioned in that because of the development of the zygote or the gametes being so different from each other, then automatically you don't have the sperm mitochondria, say from the father. That's one explanation, simply because of the differentiation of the size of the gametes. Okay. The other explanation, which was proposed now almost 30 years ago, was that somehow if the two mitochondria are in the same cell, they fight with each other. So, when they start fighting, potentially there's a war, there's a cost. So must be a mechanism to inhibit one of the mitochondria from being passed on.
That is so wild. I mean, this means that some other mechanism is, by sake of evolution, is making sure that the reproduction process happens, even though that there are struggles down below the surface among these microscopic little bits. Wow, that's wild. And so have we figured out exactly for sure why, or is that just the theory?
That was a hypothesis which was popularized for a number of years. Now, one of the things that we could test if that hypothesis were true is that if there's a conflict in the war zone after the fuse, right? What we should see is that those containing the different mitochondria in the zygote that contain different mitochondria would have a lower fitness because they're fighting internally. And the ones that only inherit one of the mitochondria would be superior. So now in this system, in the yeast, because they're the same size, we can control it now. We can test that, whether there's a conflict in there. And we did not find any evidence for conflict in that the zygote inherited both mitochondrial types or include a combination of the two parental mitochondrial types, a bit from this parent and a little bit from the other parent. They are very similar in the fitness, at least in the laboratory situation that we examined.
Do you have any trouble of determining the exact ratios? I mean, sounds complicated. I can imagine that you have to count each time. Well, it's a little bit from here, a little bit from there. Is it accurate?
Yeah, so we were able to do quite a bit, not exhaustively, but essentially what we had was we have the two parents that genetically in the nuclear genome are very similar to each other, or almost identical to each other, except the mating type locus, except the sex locus. Okay, and then we introduced the different mitochondria into them, and the mitochondria's genotype are very different through a genetic process that I don't want to get into here. It's a bit of complicated.
Next time.
So we could introduce two cells that are very similar in the nuclear genome, except they're different sexes, and they have different mitochondria, and then we fuse them, and now we can look at the progeny mitochondria, right?
Okay. I got
the
you.
zygote. So basically, we did not find any evidence for the conflict. So at least in our system, I think that hypothesis is not true. Now, the third hypothesis that I have not mentioned for why uniparental inheritance has evolved is that by being uniparentally inherited, you could control the spread of selfish genetic elements to get into the mitochondria. You know, and we all know that our nuclear genome is just full of selfish DNA, a lot of junk, transposable elements, the DNA pieces that like to replicate themselves at the expense of the host genome integrity, etc. So those could be derived from viruses, could be derived from DNA that are able to replicate themselves in there. That just, they can jump around.
Yeah.
And indeed, most of our genome is not coding for any protein. Okay, that applies to almost all eukaryotes. There's a lot of repetitive DNA in there that is not really, we should say, not essential for many of the metabolism.
Yeah, but you say it's not beneficial for us in the sense that it comes from some virus or disease that's been passed down generation. Right. Okay.
Yeah, so they collectively call them selfish DNA.
Selfish.
Yeah, they replicate at the expense of everybody else, at the expense of the fitness potentially of the host cell.
Cool.
Okay. So we have lots of those. Now, if you look at the mitochondrial DNA in animals, including primates and insects, etc., their genome has almost no junk DNA, no repetitive DNA in the mitochondria. It's very compact. Every bit is coding for something. Every bit is very important. So at least from that perspective, we know that this uniparental inheritance has limited the accumulation of those repetitive DNA or transposable DNA or selfish DNA. Okay. And then if you look at the fungi, the mitochondrial genome varies tremendously, from the small ones of 20-ish kilobase pairs to a couple hundred kilobase pairs, whereas in the humans and animals, it's about 16 kilobase pairs.
Wow.
Okay, so what we did. Sorry, go ahead.
No, I was going to say is, do you see a great difference between, like, say, humans and like armadillos, and the amount, but is it greater in the fungi kingdom? The difference
in the animals, in insects and in mammals, they're all exactly the same size.
Oh, really? Oh, wow, that's so interesting.
But differ probably by a few nucleotides. By a very small number of nucleotides.
Wow. Not
exactly, but by a very small number. They're all around 16 kilobase pairs.
Wow.
But animals in fungi can differ by tenfold. And most of the excess in the species with large mitochondrial genomes, they just have a lot of selfish elements. They call the homing endonuclease genes that allow them to jump from one place to another and replicate.
Ooh.
So because we are now able to manipulate, we identified that the genes control the uniparental mitochondrial inheritance in fungus. It's the mating type gene, it's the sex gene. Okay. When we knock out the sex determining gene, then the mitochondrial inheritance becomes biparental. Yeah.
Wow.
So the project have either of the two parental mitochondrial genotypes, or they could have a combination, recombinants of the two parental mitochondrial types.
Gender fluid.
Yeah.
Yeah. So what benefit is that to the fungi or the mitochondria?
Very good question. So what benefit do they have? So it's interesting when we did the genetic cross between the A and the alpha cells, under stress conditions like UV radiation and high temperature, they also become biparentally inherited. The progeny have mitochondria from both parents or a combination of both. So suggests that this mechanism of control is quite leaky, in that when there's a stressful condition, they allow both mitochondrial types to exist and generate recombinants. And those recombinants quite often potentially could have, good copies of all the genes in there rather than one of the good copies from one of the parents. So allow them to recombine and create very many different genotypes.
So does it? I'm sorry, but does this mean that it's more diverse in a way to where the best of the best wins out instead of the homo? Well, I forget what you said. The homo, what the animals and insects have to do. It seems like less diversity and more just like, well, give me whatever mitochondria you got, and we'll take it.
Yeah. So over here, definitely at the population level, the progeny now have very diverse genotypes, diverse mitochondrial genotypes. So that potentially could be adaptive at the population level in that some of the combinations may be fitter in one environment, and another genotype may be fitter in a different environment. So allow them to have greater phenotypic variation in the different environments. Some of them are more adaptive in one, and some of them more adaptive in another. Sorry, I was a bit repetitive over there.
Yeah. No, I mean, you have to be repetitive when you're talking to me because I am not an academic. So you didn't answer my question. I mean, obviously, your answer was you study mitochondria in fungi because it's amazing and it's really cool and interesting, right?
So it allows us to test some of those hypotheses that could not be tested using animal system, using plant system.
So how new is the information about the wide range of the kilobase pairs for the fungi? Is this new information? Or are we just finding this out?
Well, this has been probably over the last 10, 15 years since the wide application of genome sequencing. Now we're able to assemble complete mitochondrial genomes from a variety of species. So now we're finding this big variation, especially in the last 5 years or so, last few years, when many of the exotic species that have been sequenced, now we're able to see the range of size variation. Now, here's the kicker. Okay, so when we mated the strands in the conditions that allowed them to be biparentally inherited, what we found was that there are selfish elements in here. They're called homin endonuclease genes. Those genes allow them to jump from one place to another. Okay. When we have them allowing biparental inheritance, either through knocking out the sex-determining genes or in an environment that allow them to have biparental inheritance, almost all the progeny now have those selfish elements in the mitochondrial genome. So it points out that the evolution of this uniparental inheritance was actually evolved to restrict the transmission of selfish genetic elements into the mitochondria.
This makes fungi more, superior to humans. They're smarter than us.
Yeah, there are better genetic systems for addressing some of those questions. Absolutely.
The name of this episode is Mushrooms Are Smarter Than Us. Yeah.
No, that's your interpretation.
Right. That's my job as yeah. I have to make it really spicy so that people come and want to listen. But I find it interesting, but also, it's complicated. It's complicated too. And I'm glad that you come and you can explain this to us.
But hopefully I explained it clearly in that.
Well, I tell you, we did better than we did last time. So before we go, I just wanted to ask you, I brought this up before. Is it true that there are some fungi that don't have mitochondria?
Yes, absolutely. Some fungi do not have mitochondria, and that includes one of the species that causes the human diseases is a intracellular parasite, microsporidia.
Mm.
It's a obligate intracellular parasite infecting humans. They don't have mitochondria, and believe it or not, the yeast that we typically use for making beer, making wine, and making bread during the phase of generating the alcohol that we all enjoy, they do not use the mitochondria. They are fermenting. They undergo a phase called the fermentation. Even though they have the mitochondria in there, but they do not rely on the mitochondria. They use alternative pathway to convert sugar into alcohol. And if there's oxygen in there, that alcohol would be converted to carbon dioxide. So, in fact, if you were to look at some of the strains after fermentation, look at the precipitates, like the solid stuff that are at the bottom of your fermenter. Okay, there's a lot of yeast in there. Some of them may not have any mitochondria because they did not need it. So, and then of course there are other anaerobic fungi in that they don't require oxygen to live. So collectively, the ones that do not need oxygen to live, they have no requirement for mitochondria, because mitochondria is the site of using oxygen as the electron acceptor in the electron transport chain. When the electrons are transferred along the chain, to generate this proton gradient, and then the gradient then drives ATP synthesis. Okay, and eventually those electrons will be passed on to oxygen.
Crazy.
And that would be, the stuff would be oxidized and release carbon dioxide and water, converting the glucose into those components. So without oxygen, there would be no electron acceptor for most of the species of anaerobic fungi.
Interesting.
So they don't have the need. They just use the fermentation pathway to generate the energy.
And so, but do they generate carbon dioxide?
They do. Yes.
And so do, but trees take in carbon dioxide and emit oxygen. Is that right?
Yes.
Okay.
Yeah. So that's converting sunlight energy into chemical energy.
So with climate change and too much CO2 in the atmosphere, is that going to hinder my beer in my bread?
No, it's not going to hinder that process. The concentration is still too low for that to have an effect. Okay. But certainly they are impacting the photosynthetic pathway.
Yeah. I love my bread. Every night I make a little bit of starter for the next day, and I've been working with yeast and sugars and stuff, and I'm so impressed. I'm so impressed by nature.
Yes.
Yeah. So, well, what else do you want to talk about before we go? Because I know you're a busy guy. You got your office to clean, it looks like.
Yes, I should. Yes. The boxes of materials that we exchange with other colleagues around the world. So I sometimes keep some of those boxes over there. I should probably take them down.
No, you're great.
So what I want to say is that fungi not only are interested in themselves, but they can be used as model systems to understand many biological phenomena that other systems could not be used to address, including animal systems and plant systems.
Wow. And also watch out, because they are smarter than us and they will catch up to us and they will kill us.
Yes, we eventually die, and many of the fungi, they don't die. You have heard of this humongous fungus that lives in thousands of years, right?
Yeah. Yeah, I'm always impressed when I talk with you. We always have such a good time. Come back and talk to us again when more of this like Candida aura starts popping up, and these other kind of scary pathogens that are coming. I'll always be glad to get the real scoop from you. So,
Sure, would be happy to.
thanks for coming. I want to thank Xu Jianping, friend of mine, incredible human being, professor of biology down there at McMaster University in Canada, fungi master. Thanks for coming. We'll say goodbye to everybody. Bye, everybody.
Bye-bye. Thank you.